Hey guys, welcome to your first set of notes here for chapter 20. This is basically prokaryotes. We're going to go over viruses, bacteria, and archaea in that order. And viruses probably gets the most attention in your chapter. Bacteria gets a lot. Archaea gets just a little bit there at the end. If you've taken Gen Bio with me, you're going to get a lot of repetition, but it's all good stuff. And it is in a little more detail too than Gen Bio was. So I'm going to try to get you through viruses in one video. We're probably going to go over that 20-minute mark. So let's just jump right into it. Here we go. Evolution of a disease. Things that cause disease in you and I, certainly bacteria and viruses are the two that we know the most about. Bacteria and archaea are two different types of organisms that are really unicellular. They're one-celled organisms. They don't have a nucleus, but they can divide on their own. They are a cell. They are living. They're about the simplest living thing, I suppose, that we would have. Viruses, by most definitions, are not alive. They can't reproduce on their own. They must use a host. So they're simpler than bacteria. They have no chromosomes. They will just have like a viral DNA or RNA inside of them, and that might only code for a couple of genes, really, and that's enough to make a virus. They have no metabolic structures, so no nucleus, of course, but not even simple things like ribosomes. They don't have those. Really not a whole lot in a virus. They're not alive, like I already mentioned, and in terms of things evolving and changing quickly, viruses and bacteria can change very, very quickly because you and I are very picky about our DNA. We're very picky about what DNA we allow into us. We're constantly proofreading and checking with all these really cool, complicated enzymes that are running through our DNA all the time. Viruses don't have that. When they get bundled up, whatever DNA in them is more or less, to our understanding, good enough, and they take off into the world. And so they are just setting the stage for quick change, bringing in DNA that's not theirs, introducing that DNA into other people, other animals, other bacteria, and those changes just compound. And so they are the picture of evolution, of quick evolution too, because they can go through hundreds of generations in just no time. So that's a really good example there. So they do change. And if they didn't change, we could get one flu shot and just be good for life. But they change so fast that, man, we make the best flu shot cocktail we can, and it, best year ever, you might get 90 some percent coverage. It's just they're always changing. So your book starts with HIV virus as its example of a very well-known virus. HIV causes AIDS. That's acquired immunodeficiency syndrome in humans. And HIV is human immunodeficiency virus, I think. I might be wrong about the I, but I'm pretty sure that's what that means. So we first isolated this guy in the 1980s, and we have HIV 1 and 2. 1 is the most common strain. We have to give them sort of letters and numbers for names because we cannot give them genus species names because they're not technically alive. So we have to use this. They most likely, if not certainly, evolved from a type of simian immunodeficiency virus, that's SIV, and that infects apes, especially chimpanzees. And so what we're thinking this probably looked like is SIV was mutating and spreading throughout monkeys, chimpanzees, apes, and it probably infected a human who was hunting and killing these primates for food. That's a pretty common thing in developing countries to kill and butcher primates as food. And if you've ever cleaned your own wild game, rabbit or deer or fish or whatever you're catching and killing in the field, it's a bloody process, and you can cut yourself, and you're most certainly cutting the animal you killed. And so it's possible to transmit a virus from the animal you are butchering to your blood. Now, you and I are not very close to deer or fish, and so the likelihood of a virus existing in deer that would be readily attaching to our cells is not very likely, but we share a lot of DNA with primates, and so virus jumping from a chimp to a human is doable. And And sure enough, that's really what we think it did. And 1959, we get our earliest evidence of HIV in humans. Doesn't mean it wasn't around earlier, just means that's when we see it. And that's really not that long ago. If you think about it, 1960s, I mean, I know you guys are pretty young, but the 60s, it's not that long ago for HIV to be as prevalent as it is today and to have everybody know about it and for it to be such a huge problem. It hasn't been around that long. So we're thinking it probably made that jump in about the 1900s. We became aware of it, 60 years later, and now everyone is aware of it. And it's spreading, especially in developing countries, at just crazy, crazy rates. So here's what we do. We look and we sequence the genes, and we can track the movement of HIV out of Africa. And so we can get the virus out of people from Africa, we can get the virus out of people in Haiti, we can get the virus out of people in America, and we can see how it's changed a little bit. And so we think that the virus was probably carried to Haiti, by a person, I'm sure, traveling, in the 1960s. And as it's passing from person to person in Haiti, primarily through sexually transmitted disease, but certainly it's bloodborne. So, bloodborne pathogen, it can make it through blood as well. It's going to mutate. It's going to diversify in many, many hosts of people in Haiti, and it only takes one or two of them to travel to the United States. They get to the U.S. Really only about 9-10 years later, and it's there, and then pretty much it spreads quietly throughout the U.S. without us taking too much of a notice till about the early 80s, and it becomes mainstream news, and then we kick into a whole bunch of research about retroviruses and how we can stop them or slow them or keep people alive longer with AIDS and that kind of stuff. And really from that little jump from the 60s to now, we're about 30 million people who are infected with HIV today, and because it's a virus and not a bacterium, there's not a cure for it. There's not an antibiotic for it. Once you've got it from life. And you're going to die from AIDS. You're going to die from a disease that you should have been able to kick, but you can't because you're immunocompromised. And that might be 20 years from the time you catch it. It might be 45, 50 years from the time you catch it, but that's a guaranteed deal. There's no cure for that thing. That's going to be the end of you. So do your best not to get that. 20.2 is viruses and viroids, which is what we've been talking about anyway. So even though we didn't know about HIV until about the 1960s or so, we've known about viruses for quite a long time. Not a lot about them necessarily, but the 1800s, I mean, that's a long time ago. And we discovered viruses, and we use the word virus because it's Latin for like something weird like slimy poison or liquid poison or some junk like that. And basically, this researcher was able to get the leaves from tobacco mosaic, diseased leaves, and strained them through a filter that was so small it would catch bacteria. And all he got was the liquid. And when he applied the liquid to leaves, it still made them infected. And so he knew it was something smaller than a bacterium that was making the leaves sick, but he didn't know what it was because our microscopes were not anywhere near being able to take pictures of viruses. And so he just assumed that the liquid was something the bacteria made, which is pretty logical. And he named the virus, the liquid, basically. And now we know that they're much more complicated than a liquid that a bacterium produces, and in fact they're not really produced by bacteria at all in most cases, although they can be, not intentionally because the bacteria is being hijacked. But now we know they're viruses. They are non-cellular infectious particles. That's all they are. They are a little packet of DNA or a little packet of RNA wrapped inside a protein. It's incredibly simple, and they can only replicate inside a living host. They absolutely need a cell because they need that. They need the nucleus, or they need polymerase, RNA polymerase, DNA polymerase. They need something. They need vesicles. They need things, machinery, and they don't have that on their own. So they just take over someone else's machinery, and they can do that for bacteria. They can do that for a human. And really anything in between. Plants, whatever. They can parasitize essentially anything. And so our viral structure, if you're a free-floating viral particle that can infect, whatever you're going to infect, we call that a virion. And the viral genome is what's in a virion. The viral genome, it could just be a couple of genes, could be a few more, are enclosed within a protein coat. There's protein around genes, and we call that protein coat the capsid. And that's really all they are. And they can be circular, or they can look like a a cylinder. They can have like 20 sides, like a crazy soccer ball, all sorts of different stuff. And so all the capsid does is it protects the genome. You don't want your DNA or RNA just hanging out. You want it to be a little bit protected, and so they just wrap it in protein and poof, send it into the world. With any luck at all, it'll land on a host. If it just lands on like a table or a floor, the virus will more than likely die. But if it lands in a host and its membrane proteins match their membrane proteins, the cell of the host will bring the virus in, and then the virus can go about whatever its strategy is, which we'll talk about lytic and lysogenic here in a minute. The genome can be DNA or RNA. To our knowledge, we don't have any that have both. You have viruses that have DNA only, and viruses that have RNA only, but to our knowledge, there's no mixing of these two. So, the capsid consists of protein subunits, not surprising because that's what makes up protein. They assemble in a repeating pattern. Some can be helical, some can be many-sided shape, like I said, crazy soccer ball. And then some component of that viral protein needs to be able to bind to the proteins at the host's cells. If the cell proteins in your body don't match the surface proteins of a virus, you'd be very lucky and you'd be immune to that virus. That virus literally cannot infect you. If your proteins are a good match or a close match to the viral proteins, it can dock, if you will, on your cell, and your cell will just bring it right in. And so plant viruses, a lot of times, the top of a leaf is protected by a waxy cuticle, and so they need something like a wound maybe in that leaf by an insect that chews on the leaf, or maybe you're out trimming a tree or pruning a tree, some other injury where that virus can get into the plant. And so that's what plant viruses need. And of course, for humans, we just inhale our viruses right from the air, or they are sneezed onto us from our neighbor. Here's tobacco mosaic virus. Of course, the plant leaf, instead of being a consistent green, is all kind of blotchy and unhealthy. And this is the structure of the tobacco mosaic virus here, and it has a circular RNA. And then here's all of its protein subunits that need to somehow get into that plant. And when they get into the plant cell, they release that RNA. The RNA is copied by the cell's machinery, which makes more of the viral RNA, makes more of the viral proteins, assembles them, and then sends them out to infect the cell's neighbors. And so viruses don't just infect plants and animals. They will even infect bacteria, which is kind of cool because we can learn a lot about viruses and bacteria and evolution and the production of all sorts of products when we look at how viruses attach to bacteria. And so viruses that specifically infect bacteria are called bacteriophage. Phage is like to eat, so the bacteria eater, the bacteria killer, is a bacteriophage. That's a virus. And they will pierce the bacterial cell wall and the cell membrane, of course, and just inject their DNA into the cell. They have a really cool structure with like little landing legs and like this needle thing in the middle that stabs the bacterium. There are also another major class of viruses, adenoviruses. These are going to be the most frequent cause of the common cold in humans. And, the reason we don't have a vaccination for the common cold or any magic cure for the common cold is because it's caused by many, many, many different types of adenoviruses. And all of their genetic material is different, and all their surface proteins are different. And so when it comes to the common cold, you just kind of grin and bear it because there's not a lot we can do about that. These guys have no envelope. Some viruses have an envelope that is created when they leave their host. They wrap a little host membrane around them, makes them a little tougher. They can survive a little longer outside of the host. They might even be able to communicate better with the cells of the next host they attack. But for adenoviruses like the common cold, they end at the capsid. There is no envelope there for them. And so if you sneeze the common cold on the tabletop in the classroom and no other kiddo touches it for a few hours, that virus may very well dry up and no longer be able to infect you. Viruses don't last as long as, in most cases, as bacteria do. Bacteria can really last quite a long time. And so most of the animal viruses that can really cover a lot of ground, cause us a lot of heartache, are enveloped viruses, of course. If you have an envelope around you, like I said, you're a little bit tougher and you're a little bit more virulent in some cases. So the layer of the membrane that we derive from the host, the cell surrounds that capsid, and so then they have their DNA inside, they have the capsid, they have the host plasma membrane kind of wrapped around them, and they're in really good shape to cover some ground now. And one example of that in humans we have is the herpes virus, and that includes many different little classes or subsets of viruses. It would include certainly genital herpes, but it would also include the different type of herpes. I think it's herpes zoster, which gives you like cold sores. Same thing, and they can be passed from person to person, and they can survive for a little while outside of the body. And so here's your adenovirus, a very simple polyhedral. It's got 20 sides, like I said, crazy soccer ball. It also has these long filaments sticking out, proteins that will attach to, hopefully, its new host from the point of view of the virus. Hopefully they won't attach to the host if you are the human who's catching the cold. And inside of here, some double-stranded DNA. That's kind of cool. Here we have the electron micrograph of a herpes virus, and it has the envelope around it. So this is just the capsid. This is the envelope, and you can see that it has quite a bit more real estate when it has the capsid or the envelope around it, rather. And so it's derived from the nuclear membrane of the cell where the virus was assembled. And in the micrograph, the envelope is peeled back to reveal the protein coat. So they're trying to show you capsids here, DNA's in here, envelopes out here. DNA is the orange, capsid is the blue, envelope is the green. My apologies to the colorblind peoples in the video watching world. Okay, so the origin and the role of viruses. How viruses are related to cellular life is essentially unknown. We have some ideas, but they're really just ideas. I've got a cool video I'm going to show you that expounds upon some of these ideas. But like I mentioned in my whole opinion video, how viruses fit into the scheme of evolution is a bit of a mystery because they are absolutely dependent upon other cells for their replication. They're not technically alive, but they seem to have quite the ability to proliferate in the right conditions, and so they're certainly good at attacking cells. So maybe they evolved from cells, really lazy cells who didn't want to do their own work and just attacked other cells with their primitive DNA. I don't know. It's possible, I guess. Maybe they are the remnants of time before cells, when there was DNA but no replication, and then through time they became very adept at attacking and using other cells. That's a nice way of saying we don't know. That is the short answer. Depending on your worldview, from the evolutionary worldview, the naturalistic worldview, we don't know. So there you go. Viruses, well, here's what we do know. They infect and replicate in all organisms, all plants, bacteria, humans, and dogs, and cats, and horses, and birds, and everything we know. Viruses will infect them. Viruses, when they infect you, are a bad thing. If you've ever had the flu, no duh, that is a bad thing. They will decrease your ability to survive. They will decrease your ability to reproduce. Some of them can cause cancer. Some of them can cause death. They can have damaging effects on livestock. Certainly, you keep a lot of animals in a small space. That's how you maximize your profits in the ag industry in some cases. And when viruses get into things like turkeys, chickens, pigs, and they will just run through, a concentrated animal feeding operation and do a lot of damage to the whole operation, and they can also, of course, have damaging effects on humans. The simple explanation: viruses kill people. Viruses make people sick, and people can't go to work. And if you're young or old, or you have a really nasty virus, it can very realistically it can kill you depending on the type of virus you have. So there are some things smaller than viruses. Actually, there's a few things smaller than viruses that can cause disease. One is a prion, which we don't talk about in this class, but a prion is a oddly shaped protein, and there's nothing else in it. It is just a protein, and it will infect and kill animals. And then there's a viroid. A viroid is a little RNA that causes disease in plants, and it only affects plants. It does not affect animals. It does not affect humans. We're not entirely certain why. I do know that RNA does not survive long on human skin. We have enzymes everywhere that'll just tear it down. But these little RNAs with no capsids and no envelopes and nothing can infect plants. We discovered them in the 1970s. They're little circular RNAs, single-stranded, no duh, RNA single-stranded, you know that. They're super short. I mean, 400 nucleotides, that's, in the world of DNA and RNA, that's nothing really. The viroid's RNA does not encode for proteins, and it replicates that RNA using the plant's RNA polymerase, which copies RNA. That's just what it does naturally for the plant. Then it'll just use that to copy the RNA for the viroid. And when it's using up all of that time and energy replicating the RNA for the viroid, it's not expressing the genes for the plant that it ought to be expressing. And so, of course, the plant's not going to grow as tall. It's not going to be the shape and function that it ought to be, and the plant gets a disease that way. So even a very small molecule, like just an RNA, in the case of a viroid, or just a protein in the case of a prion, can cause diseases. Viroids, just plants. Prions, animals, maybe humans with Creutzfeldt-Jacobs, and probably plants. I don't know. I'm not sure about the plants actually. So viral replication. When viruses want to replicate, um, they use the same steps. First, you have to bump into a host cell. Viruses, they have no nose, no eyes, and no brain, so they just float. And if they float into your nose and are carried to your lungs, and they bump into a cell in your lungs, and they are the right fit, they hit the host cell, their proteins match your proteins, then they're in. Just by dumb luck, if they land on the road as you sneeze them out the window, then they're dead. That's just luck. That's the way it works. So then the virus will enter the host cell if it's a good match, and it's going to take over the genetic machinery. So in some cases it could just be an enzyme, other cases it's the entire nucleus or part of the nucleus, but they take over the cell. And the cell's not going to make cell products for you. It's going to make products for the virus in most cases now. And so the cell puts aside the normal tasks that it ought to be doing, and it goes ahead and makes copies of viral genes. It expresses viral genes because in the process of expressing the viral genes, it's building the viral proteins. The viral proteins are made to build the new capsid. So you copy the information, you express the information, you make the new proteins, you make the capsid, you put the copies of the information inside the capsid, and you have successfully made one baby virus. So you do this 1,000 more times, then the cell bursts open. We've made new virions as we put these new information in the brand new proteins. And when the cell bursts open, all these brand new virions escape into the environment and they float around to other cells in your lungs, or you cough them out and they float into the cells of the lungs of your neighbor in class. And that is how viruses spread, and that is how they make new viruses. And so you'll see this. They always put them in 5 steps, and sometimes the naming kind of is a little bit different. But step 1, virus attaches to the cell. Step 2, penetration. The viral particle, or sometimes just the DNA, goes into the plasma membrane. Replication and synthesis, as we make copies of the DNA and RNA, and we make their proteins. Assembly, as we put the viral DNA inside the viral protein, and release is the brand new viruses leave, and they're off to infect some other person or animal. So there's 2 pathways to do this, and this is really cool. There's the lytic pathway and the lysogenic pathway. When you and I think of someone getting sick, we think of the lytic pathway. The viral genes are expressed immediately, and it's the process I just explained to you. Lytic comes from the word lysis. Lysis means to break or to burst, and so the goal is to break the cell and spread the viruses. This is active infection, make you sick, and make as many viruses as we can while we can. The second option, which I think is way more fascinating, is called the lysogenic pathway. The lysogenic pathway is not nearly as aggressive, but it is so much smarter. The viral DNA gets integrated into the host genome. It's in your DNA and you don't know it. The viral genes are not expressed, so we don't copy their DNA right away. We don't make their viral proteins. We don't kill the cell. We're just going to leave it. And the cell remains healthy. And when that cell goes through mitosis and reproduces, it copies the viral genes and makes another cell with viral genes. And so that viral DNA is copied and passed along, and you still don't know you're infected. And then 2 cells have it, 4 cells have it, 8 cells have it, 16 cells have it, and this virus is spreading without getting the host's immune system on high alert. It's very, very cool, and it is very, very sneaky, and it hangs out in your body until later. And then someday, when you are very stressed, or you're sick with something else, or it decides this is a great time to attack you because your immune system's not on the up and up, then it can go ahead and enter the lytic pathway, make a whole bunch of viruses, and really mess your world up, which is pretty cool because I had chicken pox as a kid, which means that right now, in my body, I am passing on millions of copies of the shingles virus, which was the chickenpox virus. And it's just chilling in the lysogenic pathway, not doing a darn thing to me right now. I'm in great shape. I feel great right now. I could use a cup of coffee, but I'm healthy. And it's just going to chill. Chickenpox is in me. It's all over me. And it's being reproduced every time my cells are being reproduced. And it's not bugging me a little bit. And there's going to come a day where I get to be an old guy, and old shingles virus is going to say, hey, we've been chilling in old Rich here since he was 3 years old. He's getting a little old, getting a little slow. Let's do this deal. And shingles virus will come back in the lytic pathway, and there's not a darn thing I can really do about it. It does the same thing with cold sores. You got cold sores all the time. If you've ever had one cold sore, you've got the virus in you, and it's never going to leave. It is in the lysogenic pathway on your lip, and it's going to chill. And then when you're sick or you're stressed or you have school photos that you really want to look good for or something like that, like a wedding you really want to look good for, it'll jump into the lytic pathway and you'll get this big squishy red cold sore on your lip and you'll be very frustrated. And then it'll jump back into the lysogenic pathway and it'll chill for 2 months or 10 years and come back and get you later. So it's cool. Viruses can be blatantly obvious that they're there or very, very, very sneaky. And I just think that is so neat. So here you go. Option A, the lytic pathway. Virus particle is a good fit for the particular bacterium, so he injects his DNA or RNA. The host makes a whole bunch more of his DNA, and they make his little capsids. The proteins assemble around a coat around that viral DNA. All the parts are attached to the viral coat, and he's ready to rock and roll. So we go ahead and we break that bacterium open, and we're off to attack a whole bunch of other bacteria. The other option is we insert that viral DNA. This is lysogenic, of course. And we just throw it in the bacterial chromosome, and we don't do a darn thing. We just chill. And the bacterial chromosome goes ahead and replicates because this bacterium is going to do binary fission. He goes through binary fission, and the cell divides. It makes 2 bacterial cells. The viral enzymes are removed from that chromosome, and he can jump into the lytic pathway if he wants, or he can just relax and just keep going around and around in the lysogenic pathway to his heart's content, make as many copies as he wants. So viruses are very complicated, and they're very, very, very smart, and they're very good at what they do. And because they will just take whatever DNA is around and bring it into the virus, that could be a piece of the bacteria's DNA. He doesn't know. He just brings it into the virus. It makes them very variable. When you have a disease that has a lot of variety, it's very hard to treat. And so that's what we get. And so here we have HIV. HIV is in human white blood cells, and this is what's called a retrovirus. Now, retrovirus is very cool. If your clothing or the decor in your house is described as retro, it means it is back in time, right? Bell bottoms and things of that nature are retro. They're old. They're going in reverse as opposed to going ahead, as you would expect style would do. And we call HIV a retrovirus because it goes in reverse as opposed to ahead, as we understand that the world works like this. Every biology class knows, because they are taught this, that DNA, pardon the sloppy handwriting on the keyboard, goes to RNA, and RNA is used to make protein. And everything we understand about everything works this way. DNA to RNA to protein. And viruses do. DNA to RNA to protein. You know, they just have to use someone else's machinery. And HIV does something really amazing. Truly, truly amazing. The HIV virus does not have DNA. It has RNA. And its RNA genome is used as a template, and it uses an enzyme called reverse transcriptase because it does transcription in reverse, and it will make from RNA a double-stranded DNA copy, and then the DNA gets into the cell's genome, and now that cell's infected forever. It's amazing. It's the first time we've ever seen breaking what is called the dogma, the central dogma of cellular biology. Everything we know about cells goes around this, and sure enough, that can be broken because the more you know about the world, the more complicated it becomes, and so new HIV infected cells are produced when that infected cell replicates, and it just hangs out in that lysogenic cycle as long as it wants, just like the last slide I showed you. And so the HIV drugs a lot of time will try to take aim at a few steps. One, don't let the virus attach to the cell. Two, stop the enzyme reverse transcriptase and we can slow the spread into other cells. And so that's how we get what are called retroviral drugs, which are really created in the fight against HIV. And so that's how you get that. Here's HIV. It is an enveloped virus. So here's your envelope. Here's your capsid. Here's a little bit of fluid he got from the cell when he left, and a bonus capsid. How cool is that? 2 capsids for 1 virus. You can see why this thing is so insanely tough. Here's your RNA, and that's it. I mean, that's all you need. Some proteins to bond to the next host when blood or phlegm or whatever gets into that person, and you're set. So here's the another animation. Virus attaches, RNA's in, RNA goes to DNA, DNA's in, DNA's goes through transcription and translation, more RNA, more proteins, boom, boom, new viruses. That's kind of the deal there. And so there's step 1, step 2. There you go. So if you like it with numbers, the RNA reverse transcription to HIV DNA, which goes into the cell's nucleus, transcribes RNA that the HIV virus needs. It also translates the proteins that it needs to make its super cool envelope and capsids, and then those two come together in what's called assembly and release, just like the other virus that I've showed you does that. And so let's go ahead and stop there. We'll pick it back up at 20.4, but I'm certain we're well over 20 minutes, and I think even the attention span of the most diligent student is waning after that point. So we'll pick it up at 20.4 and talk about how viruses spread. Thanks for your time.